That's
insane. That's so
I'm
insane.
glad you got the link. For joining.
Okay, it worked. Because it kept telling me it was a link for January 19th. So, and
That's not good. I will look into that. Let's see. I think maybe before, since it's 3:59, people are probably still joining, but if you wouldn't mind just trying to share screen to make sure all
Sure.
of that works.
So you have to, you got to figure this out, like because it says here Jan 23rd. Do you have to schedule a new meeting each time, or?
You can go into the when I did it last time, I would go into the settings of the meeting and I would change it before the next one. And I would still use the same link, but you can edit a meeting and change its time. I don't know what is better. Yeah, it looks like we can see your screen.
Mhm.
Very good.
Perfect.
All right. Do you mind manning this? I'm gonna check the settings and see if I can switch it before more people get.
The Mizzou with 3 Z's is really clever.
I would like to take credit for that idea, but that was our professor's idea. So,
I think we should get started and people will join. There's a huge, there's a fairly menacing snowstorm going on outside. I don't know if anybody went out. No,
No.
it's pretty bad.
First time hearing of this.
Yes, it's a pretty bad storm.
Boy.
So I think people are just trying to figure out where they are right now. But let's get started and thank you, Neetu, for agreeing to talk at the seminar, just so that to introduce you to everybody. Neetu is a postdoctoral fellow in the Sleep Research Lab at the University of Missouri Sleep which department is it in?
The Department of Psychiatry.
Oh, okay, this is part of the medical school then.
Yeah.
And you've been there a few years now, and you got your PhD from, sorry, I should have written all this down.
Oh, yeah, that I got my PhD from University of Missouri too. That was with the Department of Radiology, and then I moved into Department of Psychiatry, both with School of Medicine.
Nice. Okay, great. Well, I'm really excited for you to chat with our group and the center, because you've told me a lot about your research and it sounds really interesting. So thank you for agreeing to talk with us. And I think the floor is all yours. We'll manage all the people coming into the Zoom room and all that stuff.
Okay, thank you. And hello, everyone. Again, it's me, Neetu, and I'd like to take this opportunity to thank Dr. Karen for this opportunity to speak with all of you regarding some of the research I've done over the years. Most of the work that I'm going to describe over here is part of my graduate school, my PhD, and then some of it is also part of my postdoc. Before I get started, I want to start with a quote that says that the human brain starts working the moment you're born and never stops until you stand up to speak in public, which I thought would be pretty appropriate for our current scenario. Four objectives for my talk here. One is to provide a brief overview of the history of human brain imaging, and then an introduction to MRI. We'll talk a little bit about structural MRI and the studies that I've done using structural MRI, specifically with gray matter volume extraction and insomnia. We'll talk about functional connectivity MRI in the healthy human brain under stress. That lines pretty closely with the work that you're doing over there, because we're looking at language networks too. And we'll wrap it up with a study that uses MR spectroscopy, which is measuring the neurochemical compositions within specific regions of the brain, and how we use that to look at treatment prediction biomarkers in autism. So again, this is the outline of how our talk will go today. I always like to start at the beginning. I've always wondered, like, how did people first think about looking at the brain or understanding how the brain worked? So I did a little bit of digging, and I figured out, like, probably the first pseudoscience to look at how to examine how the brain worked came in the form of phrenology. So it's basically measuring the bumps and dips in your skull to decide what kind of mental faculties you had and what your character traits would be. It was at one point, it was so popular, like people would try to identify psychopaths based on their skull morphology. Eventually it got disproved, of course. Then came the human circulation balance. So as you can see in this diagram over here, it's a very, very sensitively calibrated table that the patient is made to lie on. And it literally weighed our thoughts. So the brain would need more blood when it is engaged in some kind of activity. And so the bed would tilt to the side where the blood flow was higher. So a lot of very precise calculations and calibrations were required there. Then we moved on to ventriculography or pneumoencephalography with the advent of x-rays. So the x-rays helped us see the bones, and they're like, how do we get it to show us the brain? So believe it or not, they drilled holes into the skull, drained out the cerebrospinal fluid, replaced it with air, oxygen, or helium. And how they did that is by putting the patient in this chair that was tilted 180 degrees. So they're upside down, so air would go inside their brain, and then they would image the brain on an X-ray. Very creative, but had its own set of risks and problems. So some really smart people came up with the idea of magnetic resonance imaging. Very briefly, the principle is that our body contains water. Water has protons in it. In our normal day-to-day life, we don't have a magnetic moment because all these protons are aligned in random orientations. But when you put them in a strong magnetic field, they'll all align towards the magnetic field. And then you can disturb that alignment using radio frequency waves. And depending on what kind of tissue or CSF that the proton that we're imaging is present in, its relaxation after that, after being disturbed with that radio frequency wave, the relaxation varies, and that is mapped onto that signal is acquired and processed to give us a picture of the brain. There are multiple modalities we can look at within MRI. The ones we're going to focus over here, like I mentioned at the beginning, are structural MRI, which is again a detailed 3D anatomical map of the brain. We're going to look at functional MRI and functional connectivity MRI, which is based on blood oxygenation level dependent signal. So the basic principle behind that is that when one brain region is active more than the other, there's going to be more blood flow to that region compared to another. So we look at the blood oxygenation level dependent signal over there. And then the last one we're going to look at is MR spectroscopy. So it uses the same principle of magnetic moment, but instead of focusing specifically on protons, it looks at different chemicals within the brain. So what you see over here is actually a representation of within a voxel of the brain of the neurochemical compositions. So first couple of studies we're going to look at are all based on structural MRI. And this is a study that I did as part of my postdoctoral fellowship. We looked at sleep disturbance, anxiety, and gray matter volume in pain processing regions of the brain. To give you a brief background about the Mizzou Sleep Research Lab, we look at cognitive behavioral therapy for insomnia across different populations. That's one thing about sleep. It applies to all patient populations. There's some form of sleep disturbance or the other. So over here, we looked at chronic pain populations, and we were looking at how CBTI helps not just insomnia symptoms, but chronic pain symptoms as well in patients. So this is a secondary data analysis from a larger clinical trial in patients with fibromyalgia and comorbid insomnia. And insomnia is very, very, very common in patients with chronic pain, including fibromyalgia patients. Fibromyalgia is a condition where there is chronic widespread pain, but you have to have it diagnosed using something called tender point testing. So there are 18 tender points that they've identified across all over the human body. If you have significant pain in 11 out of 18 of those with a force of less than 4 kilograms per meter square, then it can be classified as fibromyalgia. And the reason I'm explaining that is because the next study is going to be about chronic widespread pain, which is different, has a different criteria. So we knew that amygdala has a role to play in pain processing. We know that amygdala has a role to play in arousal. And another common feature that we see in patients with chronic pain and insomnia is anxiety, a sense of fear with regards to their condition. So we know the amygdala is related in all of these, but no studies had so far examined exactly how sleep interacts with anxiety and gray matter volume in this chronic pain population. So that was the aim of the study, to explore the role of sleep in the interaction between anxiety and the association between anxiety and amygdala gray matter volume. We had about 50 participants for this study. They're all older than 18 years with 6 months of fibromyalgia and 6 months of insomnia symptoms. And that is chronic insomnia. And insomnia was defined by having at least more than 15 minutes of sleep onset latency, which is how much time it takes for them to fall asleep after they've gotten into bed. It had multiple awakenings, at least 4 awakenings throughout the night, and also a lack of sleep that affects their day-to-day functioning. We looked at their state or trait anxiety inventory to measure their anxiety. Then we had sleep variables, which I will go into in a second. And then we also looked at gray matter volume of the amygdala. To run the analyses, we did moderated regression models, which I will talk about in a minute as well. The sleep measures, like I mentioned, are sleep onset latency. So the patients filled out 14 days of sleep diaries where they noted down the time that they went to bed, what time they were falling asleep, how many times they woke up, how much time they were awake for, how much time did it take for them to go back to sleep. So using all of those and all that information from their sleep diaries, we calculated the sleep onset latency, how much time it took for them to fall asleep after they'd gotten into bed, wake after sleep onset, how much time they were awake after they'd first fallen asleep, and then the total sleep time, which is how much time they were actually asleep throughout the night. So these were the 3 sleep variables we were focused on. The gray matter volume extraction was performed using FSL. The MRI was acquired as part of their baseline assessments for the larger clinical trial. We did standard pre-processing, we skull stripped the MRI, we did tissue segmentation into gray matter, white matter, and CSF. We registered it to the normal MNI template. And then we used FSL to create masks for the left and right amygdala and applied those to each participant to extract gray matter volume within those regions of interest. For the statistical analyses, like I mentioned, we did a moderation analysis. The dependent variables were the gray matter volume in our regions of interest, and then the independent variables were our sleep scores, the anxiety scores, and then the interaction between sleep and anxiety. As I mentioned, we had about 50 participants in this study. You'll notice that we have a very high percentage of women in the sample, and that is very characteristic of chronic pain samples because for some reason there's a preponderance of women in the chronic pain population compared to men, especially with regards to fibromyalgia. So what did we find? We found that sleep onset latency moderated the association between state anxiety scores and gray matter volume within the left amygdala. So what you see here is when we found a significant interaction, we looked at the simple slopes at average, 1 standard deviation above and 1 standard deviation below average to see how the relationship actually plays out. So what the red that you see over here is 1 standard deviation below average sleep onset latency, which means that they took lesser time to fall asleep. So if they took lesser time to fall asleep, then the relationship between the gray matter volume and state and trait anxiety was such that greater anxiety was associated with lower volume in the left amygdala. If they took a very long time to fall asleep, which is which is marked by the green graph, that relationship is reversed. So you see greater gray matter volume in the left amygdala with a higher anxiety. So basically shows us that sleep plays an important role, a moderating role in determining how gray matter volume and anxiety interact in chronic pain populations. The reason why we're interested in this is because targeting sleep could potentially help us. We have evidence showing that CBTI has effects on gray matter volume in the brain. So targeting sleep could help us with restoring that balance within the brain, and as well as targeting anxiety and pain at the same time. The next study is looking at sleep and gray matter volume and physiological arousal. So this is a sample of chronic widespread pain. Fibromyalgia has a more defined criteria of you need to have 11 tender points out of the 18, and but chronic widespread pain is pain that does not meet that criteria. When you have pain throughout your body that does not meet that criteria for fibromyalgia, then we categorize it as chronic widespread pain. And if it lasts more than 3 months, it only has to last more than 3 months, unlike fibromyalgia, which is 6 months. There are multiple regions of the brain that are involved in both insomnia and chronic pain. And most commonly what we see is a reduction in gray matter volume. And one of the theories of the pathophysiology of insomnia is called hyperarousal, where the autonomic system is dysregulated and they're in a constant state of excessive arousal. And we've noted lower gray matter volume associated with higher arousal as well, but nobody's actually explored the influence of hyperarousal on sleep, and the relationship between sleep and gray matter volume in a chronic pain population. So that was the aim of the study. Again, we had participants who were 18 years or older, 3 months of chronic widespread pain and 6 months of insomnia symptoms. We had 47 participants in this particular study. It's again a secondary data analysis from a larger clinical trial. We acquired sleep measures, the gray matter volume, and heart rate variability. And I'll talk about that in a minute. And again, we performed moderation analyses. So the sleep sorry, was that a question?
No.
The sleep measures that we looked at were the same as I explained previously. It's sleep onset latency, wake after sleep onset, and total sleep time. The gray matter volume extraction followed the similar procedure that was described before, except for here we have a couple of we have more regions of interest that we looked at, like the dorsolateral prefrontal cortex, anterior cingulate cortex, and the right and left hippocampus. The moderation analysis follows a similar procedure to what was described earlier. Heart rate variability was used as a measure of arousal. So the participants underwent a 5-minute electrocardiogram in a quiet environment. They were given 3 minutes before that to acclimatize to the EKG recording. And then the time series analysis of the electrocardiogram was performed, and we computed what is known as the average root mean squared standard deviation of normal heartbeat intervals. Basically, it's just represents a measure for arousal. So if there are lower values of this variable RMS DDN, lower values of that variable represent higher physiological arousal. That means that you're in a constant state of overworking yourself. So it doesn't change with their demands of your task. For this one, like I mentioned, we have 47 subjects. Again, we have more females than males for the study. The average heart rate variability and sleep characteristics are all described in the table. And what our results suggested was that if you look at the first figure, at increased arousal, there was increased gray matter volume for increased sleep onset latency. And then at higher levels of arousal, which is the red line that we're looking at right over here, there a decrease in decrease in gray matter volume was noted for decrease in sleep onset latency. Basically, it's saying that if you fall asleep quicker, you have lower gray matter volume compared to those who take longer to fall asleep. And with lower levels of arousal, which is the blue graph right here, this relationship was reversed, and this is specifically in the anterior cingulate cortex. We also found that in the hippocampus, in the right hippocampus, we found a reversed relationship, which one of the explanations for this reversed relationship could possibly be that the hippocampus being located in the temporal lobe, it's one of the temporal structures. It is more susceptible to the effects of stress and higher arousal. So maybe that overtakes any kind of effect that sleep has on this relationship. So the preliminary findings from this study indicate there's a complex interplay between the arousal and insomnia. And like I mentioned previously, cognitive behavioral therapy is known to help decrease arousal and increase HRV. So it could possibly be targeting multiple factors in a chronic pain population, such as not just sleep, but their arousal as well. With that, we'll move on to functional MRI. And the functional MRI studies are going to focus on stress, cognition, and functional connectivity. The first study will look at the effects of stress on functional connectivity during verbal processing. And we know that stress, long-term stress, has multiple health concerns associated with it, like cardiovascular problems, but even short-term exposure to stress or acute stress can cause difficulties with cognitive functioning and working memory or executive functioning. And when you are in a state of stress, it leads to heightened arousal, and that results in a more narrowed, focused attention. So it's almost like your fight or flight response is being triggered. So you are trying to figure out because stress is a normal physiological response to what the body considers a threatening situation. So in that event, you're always thinking about, oh, do I want to flee or do I want to stay? You don't have access. It reduces your access to multiple different remote networks to come up with a creative solution. So the more stressed you are, it's harder for you to come up with a creative solution or look across different networks to figure out an alternate solution than the fight or flight. So over here, we were focused on understanding how stress affects the neurocorrelates of how stress affects language networks. We specifically chose language networks because they involve extensive searches through multiple networks. It also has implications for specific populations, such as people who have test-taking anxiety, public speaking anxiety. So we wanted to see if functional connectivity could serve as a marker of sorts to target for intervention to counter these negative effects of stress. We had 36 participants here who were older, 18 years or older. No history of medical or psychiatric condition, no depression, no anxiety, completely healthy. One of the factors that we wanted to look at is the influence of genetics on stress susceptibility. So the serotonin transporter gene has a polymorphism that we call the S allele or the short allele. And people with that polymorphism are known to, in literature is described that they are more susceptible to the effects of stress compared to people who do not have that S allele, and they're called the L allele subjects because they have a longer allele. So that was one of the things that we wanted to explore. We wanted to look at how stress affected the S allele and L allele participants differently. We also wanted to look at how stress affected functional connectivity across gender as well. So for the study, the participants underwent fMRI acquisition and verbal fluency assessment, which I'll pull up in just a second. The data analyses involved repeated measures, MANOVA tests, looking at differences in activation, differences in average functional connectivity across all regions of interest, and then functional connectivity strength between specific individual ROI pairs in the stress and no stress condition. So this is the protocol that we used within the scanner. The MIST is the Montreal Imaging Stress Test. It is a adapted version of the Trier Social Stress Test specifically for use within the scanner. It involves a range of mental arithmetic tasks of increasing complexity. While outside of the scanner, participants are made to do the arithmetic tasks, and then within the scanner, it's programmed so that they only get an accuracy rate of 50%. And for each response that the participant indicates, they get socially evaluated feedback. So they'll say, oh, that was 90% got this answer right. Oh, you need to try harder on that one. This one isn't, that wasn't the correct answer. And sort of social evaluative feedback that is meant to induce stress. So that was the missed block that was used to induce stress. And then right after that stress induction, they were asked to name as many animals that they can think, as many things that they can think of that belong to a certain category, like animals or things to wear or food. That was the categories task. And then we repeated this with a letters task as well, where they were asked to name as many things as they can think of that would begin with a certain letter. And we had 2 runs each of the categories task and the letters task. We looked at specific regions of interest that were known to be implicated in language processing. So we had the we have the left inferior frontal gyrus, we have the left middle temporal gyrus, the left parietal lobe, and the left fusiform gyrus, which are all involved in different aspects of language verbal processing. So for the functional connectivity analyses, what we began with brain extraction, and then did something called slice timing correction, which is dependent on the kind of machine that you acquire your images from. We did motion correction so that any head movement that was more than 2 millimeters in either linear or rotational axes were removed from further analyses. All patient data was registered onto the standard MNI template to enable comparisons across participants. And the stimulus protocol was fed into the general linear model, and the resulting activation maps, we extracted the time series, and all of these processes were done using Excel. We applied the ROI masks and extracted the time series. We ran the correlations and Z-transformed the results in order to get a measure of the Z-score measure of functional connectivity strength. So again, this is what our sample looked like. It was 36 participants. We tried to get as equal a distribution of S allele and L allele and male and female as we could. But again, we had a preponderance of females, almost like twice as many as males. There was no difference in terms of their IQ. And what you see over here is the group activation masks for when the participants were performing the letters fluency task under no stress. So we had, I'm sorry, I should have mentioned that. We had a counterbalance design. So they came in one day and they had the stress paradigm, and then they came in with a gap of at least 48 hours. And they did a no stress paradigm where the same tasks were repeated. And instead of the missed, they just had to sit there and count 1 to 10. The results of the study indicate that there was increased functional connectivity between the left inferior frontal gyrus and left fusiform gyrus, as you can see in this figure right here. And we also found that the S allele participants were thought to have greater stress susceptibility, had lower strength of connectivity between the left inferior frontal gyrus and the left middle temporal gyrus. So it appeared that stress not only added more load to long distance connections, like the one between the left inferior frontal gyrus and the left fusiform gyrus, but within in a genotype specific manner, it also affected short distance connections. So as a result, we deduced that stress had effects on, and we also found that these differences in functional connectivity were associated with their performance on the tasks. So it also was correlated to how many words they were able to produce that belonged to a certain category or a certain letter, began with a certain letter. So that indicates that functional connectivity could be used as a marker of how stress affects performance on cognitive tasks such as language tasks. The next one is effects of stress on functional connectivity during problem solving, creative problem solving. So there are mainly two types of tasks that are involved in creative problem solving, or convergent tasks and divergent tasks. Divergent tasks are open-ended, if you will, in the sense that they don't have one correct answer. You can come up with anything you want to, depending on your level of creativity. It's like saying, come up with an alternative use for a pen instead of writing. So it can have any number of responses and they could all be correct. But in convergent creative tasks, you have one single solution to the problem, but it requires searching through multiple remote networks to come up with an answer that might not be as obvious. So you have to find an answer that is, you have to suppress the more dominant response that you get and search for more for answers that might not be as obvious. And then we've noticed that high levels of creativity are associated with higher levels of functional connectivity. But again, acute stress affects this ability to access remote, less obvious networks under stress. So we wanted to look at how functional connectivity based on gender and genotype changes under stress for a creative problem solving task. Again, we had participants that were 18 years or older. There were 32 participants for this arm of the study, no history of medical or psychiatric condition. They were all in good health. We did genetic screening and genotyping, similar to what was described previously. Did fMRI acquisition and verbal problem solving, and the data analyses followed a similar pattern to what was described previously. For the verbal problem solving task, we used what is known as CRA, the Compound Remote Associates task. So it is basically, they're presented with three different words, and they have to come up with a target word that forms a compound word with each of those three. So as a very random example, it can be bee, tree, and suckle. Oh no, bee, tree, and honey, maybe. And then there'd be just one word that goes with all three of them. For example, apple, so like apple bees, apple tree, honey apple, and that's just a very random example. But there is a very specific answer to each of those combinations presented to them. And then so we followed the same paradigm of having them undergo the stress using the MIST protocol. Then they were presented with the CRA. They had 6 CRA problems within each task block, and then they were given that they had to do, then given a rest period, repeat the MIST, repeat the CRA. And then when they came in on a different day, instead of the MIST, they would just do a regular counting 1 to 10, and then do the CRA without being stressed. Again, we had specific regions of interest that we wanted to look at, that we knew were involved in language processing. And the fMRI functional connectivity processing pipeline was pretty similar to the previous study as well. What you see here are group activation maps for participants performing the task under stress and under no stress. Clearly, under no stress has more activation than under stress. What we did find was a significant stress-gender interaction in the right superior temporal gyrus. So the right superior temporal gyrus is involved in verbal creative tasks and also maintains solution-related activation to problems that are not solved yet. So if I was presented with one problem, I couldn't solve it, and I moved on to the next one, there's a part of my brain that's still thinking about the problem that I did not solve. So it appears that in males, activation in that area is higher than females in the stress condition compared to the no stress condition. And in terms of functional connectivity results, we have a three-way interaction effect between gender, allele type, and functional and stress condition. So S allele males had decreased functional connectivity between the left inferior frontal gyrus and the left middle temporal gyrus. And the L allele males, on the other hand, had increased functional connectivity in that same region compared to the L allele females. So it's a rather significant difference. We also found that the connectivity was for S allele individuals, the functional connectivity was positively correlated with performance under stress, but for the L allele individuals, their functional connectivity was negatively correlated to performance under stress. So if they had higher strength of connectivity, it meant they performed worse on the task. So again, we specifically with the left middle temporal gyrus, we seem to have a marker, connectivity marker for populations with susceptibility to stress. But then because the sample size of the study was rather small, and this is the three-way interaction, so within each group, the number of subjects was pretty small. We cannot make a definitive conclusion yet. We're going to have to repeat the study in a larger sample in order to be able to make those definite conclusions. And with that, we'll move on to magnetic resonance spectroscopy. So this is a very small pilot study that we did to see if we could predict pharmacological treatment response in autism spectrum disorder. So autism spectrum disorder is behaviorally defined complex disorder. It's early childhood onset, and it has three main hallmarks, which are difficulties with social skills, trouble with communication, and also the presence of restricted repetitive behaviors. So right now, we do not have any pharmacological interventions that are approved, FDA approved for the core symptoms of ASD. The only treatment they have is for irritability associated with the condition. So in terms of finding a treatment for the core symptoms of ASD, we went back to look at what's implied in the pathophysiology of the condition. We found that literature repeatedly refers to an imbalance in excitation and inhibition within the brain. So the excitation is governed mainly by glutamatergic neurons within the brain, and inhibition is governed by the GABAergic neurons in the brain. So you'd have an excess of excitation and not enough inhibition to be able to function properly. And in postmortem studies, these imbalances in excitation and inhibition were consistently reported in the dorsolateral prefrontal cortex and then the cerebellum in ASD. So we looked at a drug called memantine or Namenda, which is a glutamate antagonist. So they have an excessive glutamate activity going on in their brain. So, and we looked at a drug that is a glutamate antagonist to see if that would help people with autism. There have been random case studies with Namenda before, but nobody had done a clinical trial with it yet. The random case studies all yielded mixed results. So we wanted to see if glutamate levels in certain regions of the brain, like the dorsolateral prefrontal cortex and the cerebellum, would predict how a participant would respond to treatment with Namenda. So we had 10 participants, all above the age of 16. They had an ASD diagnosis that was per the DSM-5 of or ADIR, which is the Autism Diagnostic Interview revised, or the Autism Diagnostic Observation Schedule. And for this particular study, we only looked at patients who had mild to moderate illness. That was quantified by a score of less than 4 on the Clinical Global Impression Severity Scale. So the Clinical Global Impression Severity Scale is a clinician-rated scale. It goes from 1 to 7, and 4 means whatever feature that we're looking at is not as is not that severe. And then any 1 to 4 is the mild to moderate range, and then 4 to 7 is the severe range. The reason why we picked the mild to moderate range was that we were going to put these participants in an MRI scanner. So with a more severe level of autism, it is harder to get them to lay still in the scanner to perform that scan. And also for other parts of this clinical trial, we had behavioral assessments that we needed to perform that were not conducive for patients who had severe autism. The measures we looked at were the clinical global impressions. We also had something called the social responsiveness scale. Like I mentioned, we were interested in targeting the core symptoms of autism, and one of them is a deficiency in social skills. So the social responsiveness scale was one of the measures we looked at. And we also looked at MR spectroscopy derived neurometabolite concentrations for glutamate and a bunch of other metabolites in the dorsolateral prefrontal cortex and in the cerebellum. For the analyses, we ran independent sample t-tests, Pearson correlations, and stepwise linear hierarchical regressions. So this is an approximate position of where the voxels were placed. So there are two types of MR spectroscopy. There's single voxel and there's a whole brain MR spectroscopy. What we used here is a single voxel. So a single voxel of 2 by 2 by 2 dimension was placed in the left dorsolateral prefrontal cortex or the right cerebellum. And we had the same person place the voxels each time for each participant to avoid any inter-user variability. So what we found was this is the score on the social interaction scale post 12 weeks of treatment. So this shows their improvement. Like I mentioned, 1 to 4 means an improvement, and 4 to 7 means worse or more severe. So all of our participants showed some form of improvement, except for 2 who withdrew from the study because of negative effects of the drug. So they haven't been included in here because they did not complete the whole study. And but the rest of the participants did have show improvements of varying degrees or no worsening of symptoms. Unfortunately for us, we didn't find any significant effect of glutamate levels in the dorsolateral prefrontal cortex or the cerebellum on between responders and non-responders. So in this CGI, based on the CGI score, all of the participants were classified into responders and non-responders. And the score of 1 to 3 was the responders category, and the score of 4 to 7, which is no change to worsening symptoms, was the non-responder category. So we didn't see a difference in glutamate levels. We did find significantly higher levels of NAA, which is N-acetyl aspartate, in the left dorsolateral prefrontal cortex in the responders compared to non-responders. Then we looked at how that correlated to the behavioral measure, which was our social responsiveness scale. And the change in a social responsiveness scale was negatively associated with the creatine, phosphocreatine levels in the left dorsolateral prefrontal cortex. And we also went ahead and tried to come up with a model that would predict response to memantine post-treatment with memantine, and found that in the final model, the concentration of glutamate and inositol in the left dorsolateral prefrontal cortex was predictive of scores on the social interaction scale of the CGI. So a bunch of mixed results, some aligned with what we were hoping to see, and some not so much. But as a result, we think because of the interaction between the observed results, the difference in NA levels and the creatine phosphocreatine levels, we feel that the response to memantine is governed more by mitochondrial dysfunction, which is also a characteristic hallmark of autism spectrum. So it's probable that you have a subset of people who have this mitochondrial dysfunction that's associated with higher levels of NAA in the left dorsolateral prefrontal cortex and decreased creatine phosphocreatinine in that same area that will respond better to memantine than those who do not. That's one of the things that we struggle the most with in autism literature and autism research is because of the heterogeneity of the disorder, similar to what you see in stroke. Every stroke is different, same way each person with autism spectrum disorder is different. So it's possible we have like different subsets that will respond to a certain drug and a different subset that will not. So definitely larger studies are warranted to be able to understand what are the factors that drive treatment response within the ASD population. So after all this, we're talking about, we're going to come back to square one. We're going to talk about magnetic resonance imaging. Like I mentioned, it offers an exciting opportunity to look at the brain structure, function, and even composition now. So helps us identify brain states in health and in disease. It helps us study human cognition. It helps, it can be used as a treatment prediction biomarker, like I mentioned, for the ASD population. I've taken some of the aims of the Center for Brain Recovery, and I wanted to take a minute to show you how that aligns with the work that I've done so far. So clinical trials for neurological disorders. I've worked in clinical trials all through grad school and postdoc. I'm also excited to look at the role of sleep and arousal in rehabilitation processes and stroke. In terms of neuroscience of the everyday world, definitely the use of bringing in the use of more advanced technology in neuroimaging, such as MR spectroscopy, to understand the brain function in health and disease. In terms of identifying biomarkers, again, implementing advanced neuroimaging techniques, which I've been doing so far in terms of determining functional connectivity, in terms of using MR spectroscopy to figure out biomarkers of treatment, response to treatment. And data science and artificial intelligence. I spoke to Dr. Kieran earlier, and I realized that my understanding of that was a little different from what she had in mind, but still, because neuroimaging data sets are so massive and huge, it's an amazing opportunity, I think, to be able to implement machine learning and automated algorithms to perform neuroimaging analyses. With that, and this is disclosure, funding for the different projects that I talked about came from the National Institute of Nursing Research, the University of Missouri Research Board, and the University of Missouri Mission Enhancement Fund. I would like to thank my mentors, colleagues, lab members, and all the participants for all the studies without whom none of this would have been possible. And I take any questions you have. Thank you.
It might be easier if you stop sharing your screen unless somebody has specific questions. You can always go back to just sharing your screen unless it was too hard to do that. So we're all online, so it's easier to probably ask you questions this way. Where do you want to start? Can I start with a question about spectroscopy? I don't think I fully understand how that works. So how do you collect the data, and then how do you collect the data about these creatinine levels all over the brain, and then you decide to only look at specific regions, or do you collect that information only in certain regions of the brain?
We collected that information only in certain regions of the brain. So we use single voxel and wherever we place that single voxel, you can extract information from that specific area, but you can do a whole brain as well.
But in terms of collection, you can do the whole, like you can go around the brain and look for these values.
Yes, you can do a whole brain analysis with it's a much more the processing is much more time consuming, but it can be done.
And in every given voxel, there's a sort of comparison of what is considered to be healthy control, healthy data as a comparison.
So it used to be the case that when they first started off using spectroscopy, all the measurements were performed relative to creatinine, phosphocreatinine, because they thought that was going to be uniform throughout the brain across people, but that is not the case. So we have specific frequencies. I'm just going to quickly go back and show you that graph again to explain it better. No. We do have specific, we look more at the spectrum than at the actual value, if we will, and that'll be more clear when I show you the picture. Oh. Yeah, so if you look at this, this is the resonance frequency. So NAA is obviously most abundant in the brain. So that always comes at a certain level above the rest of them. So it's almost like a comparative ratio than saying that exactly this is how much NAA is present in a healthy human brain compared to one that's not. Does that make sense?
Yeah, I think I understand that now. Thank you.
No problem.
Other questions?
Can I ask you quick questions?
Sure.
Yeah, let me just turn on the video. Yeah, my name is G, and I have quick question about your study on sleep disorders, and the age range seems to be pretty wide, from teenagers to up to 70s, and I read some of the literature which suggested that sleep disorders is associated with the symptoms of the mild cognitive impairments, and all the symptoms of the cognitive impairments in the aging process. So in your data set, how you controlled the aging effects in your analysis? That's one of the question. And number two is how you think about this sleep disorders is, the relationship between sleep disorders and cognitive impairments.
So those are really good questions. First of all, like I mentioned, this is like a secondary data analysis from a larger data set. When we were recruiting the participants, we did a battery of cognitive tests in the beginning to make sure we were none of them had any kind of cognitive decline. So they're all cognitively well, if you will. And then there is a definite relationship between sleep and cognitive decline over the years. So people who have insomnia, chronic insomnia, have a higher chance of developing Alzheimer's than people who do not. So one of the trials is there are a number of trials that we have ongoing in our lab right now, and one of them is specifically focused on targeting people with mild cognitive impairment, bringing them in early on, treating them for insomnia, and following them longitudinally to see if it delays their progression into Alzheimer's. So it definitely is there. Part of it is thought to be through the gray matter volume reduction that's observed in the brain because of chronic insomnia. So that's one of the mechanisms that has been proposed. But a deeper understanding, we'll have that only after, hopefully, our large clinical trial gives us amazing results.
Thank you.
No problem.
You just have to wait a little bit longer at 5 o'clock for people to unmute themselves. So I'm just gonna wait a little longer.
Yeah, completely fine.
Yeah, if there are no more questions, can I, oh, if there is,
There seems to be an online question. Can you read that?
Okay. I think it's Vinod Jagaru. It says, apologies for not turning on my video. I have cadavers behind me in an anatomy lab. In terms of amygdala activation or dysregulation seen in your studies, were they seen uniform across the entire amygdala or weighed in one direction, example, the basolateral or the corticomedial?
Sorry, I can clarify. I apologize. Yeah, I meant uniformly. Yeah, if we can just clarify that where they seen.
Sure, and that's a really good question. Thank you for asking that. But we looked at uniformly across the amygdala. We did not separate into basolateral or mesolimbic aspects of the amygdala.
Okay, I do have a somewhat unrelated follow-up. You know, as you probably know, the Yerkes-Dodson law done in rats in early 20th century suggests that there's an optimal balance between arousal states and performance, and that famous upside down U curve.
Yes.
So how do you calibrate? And this is purely curiosity question. How do you calibrate your experiments to determine where's this optimal level of performance? And you know, if someone were to, what does optimal mean when you're looking at MRI data, the functional MRI data?
So that's again another really good question. Just to give a bit of background, there is an inverted U. So stress is supposed to help us. It's designed to help us make the best out of our situation. So that is why we have hyper-focused attention onto the problem at hand, and we try to solve it however we can. So if I'm understanding the question correctly, it's like, how do you determine when the balance goes from helping us to being detrimental to us?
Yes, that's correct. Yes.
And I think that is reflected in the behavioral assessments that we do. So just not being able to perform, we are comparing them after they did the MIST protocol. They were under stress, and then they did a bunch of cognitive tests. And then we're comparing it to when they were not under stress, and they did a bunch of cognitive tests. And we're seeing how their performance varies in both those conditions. So I think that gives us an idea of whether the stress induced was severe enough to be detrimental for them, or was it not? And which in our case, we did see definite effects of the stress on the cognitive performance as well.
Okay, great. Thank you.
You're welcome.
I'm watching the time. Unless there are any other questions, Neetu will let you go. I know it's been a long day. We've kept you busy since 9 o'clock. So thanks a lot. And thanks everybody for joining today. It was great. It was a great presentation.
Thank you so very much. I really appreciate the opportunity. Thank you very much.
Bye.
Bye.